Good afternoon and welcome to the FoodBall Data Analysis webinar. I hope you can all hear me okay. If you cannot hear at this stage, if you could write down in the chat at the bottom of your screen, and then we can try sort something out. So we're going to start today with presentations from Carl Brunius and from David Wishart. So the objective really of this webinar is to cover some data analysis strategies for time series data. So it's not to force you to do your analysis in any way using these methods you're going to hear about today, but rather it's to give you some options or some flavor for the type of data analysis that you could perform. So as I said, we've got two speakers. They both will speak for about 20 minutes, and then we will have time for questions. We will have questions by typing in questions on the chat button at the bottom of your screen. So if you have questions as you're going along, you can type them in, and at the end of each presentation we will have about 10 minutes to ask questions to everybody. So let's hope this works well, and with that I can hand over to our first presenter, who is Carl.
Thank you very much, Lorraine, and good evening one and all, except for the Canadian guests for whom it's good morning. My name is Carl Brunius, and I share my time between two universities, Chalmers University of Technology in the south of Sweden and the Swedish University of Agricultural Sciences. And my job is to develop tools for multivariate data analysis, and of course to apply these in settings predominantly related to metabolomics. And I'll just share with you today some overview of statistical methods and a series of some data that we have been working with, some approaches that we have tried based on an over decomposition and multi-level analysis. Try to wrap that up and conclude and give some kind of outlook, what we sort of think to be the direction for the future, at least in our lab. So, I mean, this is not an exhaustive overview of statistical methods, but this is sort of a mental roadmap for me, where I tend to look at type problems in the columns and the type of data that I'm facing in the rows. So if I face some kind of situation, I can sort of choose a classification type that would be a t-test in a parametric situation, but in multivariate it's the workhorse, the PLS, discriminant analysis, of course. And depending on the type of task that we're facing, then we might want to choose a method, not limited to these, of course, but this gives us sort of overview of what's out there. And we see that there are in fact multivariate methods for most of the things that we're doing, from visualization, it's our friend PCA, to regression and classification analysis, where we tend to have PLS as the de facto standard. But there is also methods like random forest or support vector machine, which are gaining momentum also in the metabolomics field, which is very nice. For the dependent two-group test, there's a multi-level approach, and for multiple factors or time series, that's been really a problematic field. But there's the sort of pioneering work of the ANOVA simultaneous component analysis, which is very interesting and promising. So we've been working on two approaches actually. One which is based on an over decomposition, and the things that you're going to see on the bottom, on the sort of red circly thing, is APLS, ARF, ASVM, which is an over decomposition but not combined with simultaneous component analysis, but rather with our friends PLS and random forest. And that's one way to look at time series data, and I'm sure you'll hear more from David Wishart about that as well, because it's also offered by MetaboAnalyst. But what we've also worked with is area under the curve from time series data, and looked at differences between treatments in a sort of pairwise multi-level analysis, and then find which variables are important, and then look at them in univariate statistic to cover time effects. All right, so just to get a feel for the kind of problem that we've been working with, it's with the University of Barcelona, the group of Cristina Andres La Cueva. They've been doing a dietary intervention and a crossover design to find biomarkers of exposure related to dietary pulses. So it was a single morning test meal of 300 grams cooked food product. So it was control which was pasta, lentils and chickpeas, and there was also an extension of the trial which covered beans. And as you see from the numbers, it's only 11 individuals, so it's fairly low power, but it's a crossover design, so I mean, it's really interesting situation. And just so you have sort of full story, that standardized other meals from the evening before up to 48 hours after the dietary intervention. And serum and urine samples were taken from these individuals at regular time points, which you're going to see here from, so it's from baseline, 1 hour, 2 hours, 4 hours, 6 hours, 12, 24 and 48 hours. And we measured this by all the samples by NMR at the Swedish NMR Center, really fantastic people to work with. So we borrowed the sort of pipeline from our targeted workflow regarding where we're using normally LCMS to work with features. So we pretty much just took the raw data, aligned it in R using the now famous Speak package, and found which were the peaks, extracted those peak heights to work with as a proxy for sort of feature intensity. And we were facing in the urine data something like 1,500 features, and in serum data we looked at 500 features-ish. And what's actually interesting with this approach is that when we extract peak heights instead of working with peak areas, we're slightly less sensitive to overlap between different peak areas. But that's something else for us to discuss in a different webinar on strategies for NMR data and metabolomics. So the first thing that we did using this data, that we got 11 individuals, 3 or 4 treatments, depending on how you see it, and different time points. It's to go to our friend ANOVA decomposition, which may seem daunting, but what it does is you borrow the sort of general idea behind ANOVA analysis to decompose the data into partitions. So we dissect the overall variability by the main effects. So we're just assuming that your data here represented by the X for individual treatment time, it's centered, it's scaled and it's centered. So the mean value would be the mu in the mathematical model in there. So the first step is if we look at this sort of naive graph with individual clusters, is that each colored blob sort of represents different data points in a PCA or something, where each blob represents multiple measurements sort of clustered around each individual, because the individual is very often going to be the main determinant. So how do we look at time effects when we have so much effects of the individual? Well, if we center around the individual, we do that by removing the individual average. And then we center all the data around this sort of origo, and we end up with a different situation in which we may find that we have new clusters of data points where the clusters may be related more to the treatments. So if we find that data partition, we can remove that variation and then look at, for example, the time trajectories. The beautiful thing here is that we can model any and all of these data partitions individually separately from each other by, for example, PCA technique, which is the fundament of the ASCA. And we can also add in the residual back into different partitions to be able to do supervised analysis as well. So we actually did this ASCA analysis, and it's been pioneered by the Biosystems Data Analysis Group in Amsterdam, and there are procedures available both in MATLAB and in R, and as well in MetaboAnalyst for doing this. So we did this on the serum data, which was what we had available at that time, and we did a PCA on different partitions to find different time effects, but we saw that there was not very much to be found there. So we tried supervised analysis of the decomposed data instead, and there was basically nothing to be found here. So we actually abandoned this approach to work with another approach that we've been trying previously. No, sorry, I'm moving ahead of myself. Just to wrap up sort of pros and cons of the ANOVA decomposition, that of course on the plus side, that we can do direct analysis of time series data to look at kinetic behavior in the multivariate analysis. This is really an upside of this type of approach, and we can use the different partitions to study different effects, for example, overall treatment effect versus overall time effect, or these things. On the con side, a lot of people find this to be a complicated or complex approach. I do myself, actually. So how to use all of these partitions, it's not always very straightforward. And in our case, the main con was that we didn't get any useful information from that. But at this point, we didn't know whether it was related to the strength of the data or lack of strength in the method. I don't know if you are seeing the correct screen or not. I'm getting these weird sort of flashy things. Anyway.
Yeah, we're seeing the screen. It just flashes every now and then, but we're seeing it the majority of the time.
Great. I don't know why it's doing this. It's a secret life of computers. Anyway, I hope you. Oh, we're seeing most of it anyway. So our second approach was to actually remove the kinetic information in the data to work with the area under the curves. So if we look, for example, at this situation for this metabolite, which we found at 0.85 ppm. Then we did area under the curve in the time frame from 0 up to 6 hours, because we figured that that's going to be predominantly the region in which we have an effect, and at later time points will be confounded by other meal effects, and it's not fair to draw an area under the curve if we have got peaks that we're actually not covering due to the fact that we lack resolution in measurements at those later time points. And this is something that we've done before in time series analysis. So basically, since it's a crossover design, we can do a multi-level pairwise partial least squares analysis on this. And this was in fact the procedure that we chose to continue with a Barcelona team, and it's Fran Madrid-Gamin who's working mostly with this data. And what we found was, from the initial results on serum, that we got nothing, basically, using the full data set. If we employ some variable selection, we can get some type of results with strengthening the multivariate modeling, but of course this results in overfitting, so it was significant, but it was not very nice. At this point in time, we received the urine data, And since we were working on the area under the curve and the multi-level analysis, we decided to continue with this, and it turned out to be really beautiful. And if you're used to seeing multi-level analysis, basically what you expect is to see in the graphs in the lower area of the screen, in lentil versus control, for example, and in the upper left part you want to have observations there, in the lower right part you want to have observations there. Anything in those other two fields would be a misclassification, and we find that we're very nicely managing to separate between lentil treatment versus control treatment, chickpea versus control, and chickpea versus lentil. So we find that in the urine data we get really nice separation. And we employed nested cross-validation to minimize the risk of overfitting, and confirmed this by permutation analysis as well. But we also did this using a sort of new validation framework, which we have been working with in our lab for quite some time, which is a nested cross-validation or repeated double cross-validation, which has also been pioneered by the Biosystems Data Analysis Group in Amsterdam and Fieldsmoser and colleagues elsewhere. So we do the same kind of thing, but instead of just tuning over the number of components, we're also tuning over the number of variables within the cross-validation loop. So we get an unbiased variable selection. So out of these 1,500 variables, we actually get a choice of 45-ish metabolite features that were selected from each model, and these are in fact the metabolites that we're going to be looking at, which are going to differentiate between the different treatments. So it's a very strong modeling, the multi-level approach, of course, and we got the automatic selection of informative variables, which is on the plus side. But on the drawback is, of course, that we don't have kinetic information in the multivariate modeling. So what we did instead was to take these metabolites that we found to be relevant and then do univariate repeated measures or marginal model, depending on your vocabulary, and this kind of modeling to find out whether the treatments are significantly different from each other, with its treatment time interaction and these things. So that's one way to look at the kinetic data. So the pros, just to reiterate, it's a fairly straightforward procedure. Each step is relatively easy to understand and perform. It's combining into area under the curve, performing multi-level analysis, and then performing repeated measures on the selected metabolites. So each step is rather powerful in itself. But on the downside, there's of course no time profiles in the multivariate modeling, and also the fact that we did not use the 12, 24, and 48-hour time points in the modeling because, well, it's really quite tricky. How do we deal with this situation? And of course, there's a development potential to do more non-parametric kinetic modeling instead of doing the repeated measures and over. But this is where we started, and we thought it produced some very nice results. But what we would like to do in the future is return to the ANOVA decomposition of the urine data. The ANOVA decomposition of the serum data was not very nice. It didn't really give us anything, but now that we have the stronger urine data, we think that we could probably find something here. So we can do direct multivariate analysis of time series, but on the downside, it becomes slightly more complex to look at the treatment time interactions in a supervised model, and we're not really there yet. There's still development to do before we can consider this a viable option. So to try and wrap up these two approaches, so there are many possible ways to analyze data. as we all know. And the most promising strategies that we've seen so far, at least in our group, is the multi-level analysis of AUC and then combining that with a marginal modeling for kinetics. So it's straightforward, but the downside that we cannot look at the time effects directly. And then there's the ANOVA decomposition, which can be done unsupervised in the form of PCA or supervised in the form of PLS or random forest. So we can look at higher complexity directly in the multivariate analysis. But how do we look at the supervised analysis of the treatment time interaction where slightly complex. So we need more development to enable us to do that. And to conclude, I just want to thank the co-workers and collaborators in this. It's of course the Chalmers SLU team, which is headed by my professor Richard Landberg and our PhD student Lynn. The Barcelona team headed by Cristina Andres La Cueva and the Swedish NMR Center. And I want to especially highlight from the Barcelona University, Madrid Gambin, who's doing most of the sort of grunt work of data analysis. So I thank you very much for your time, and I'm happy to take questions.
Thank you very much, Carl. So if you have questions, you can go down to on the bottom where you see chat, and you can type in questions there for Carl. So maybe I can just start off, Carl. With the multi-level AUC approach, did you have to correct for multiple testing? Because I presume you've done an AUC for each one of your individual features.
Yes. There's an AUC for each and every feature, but the way we do the multivariate modeling, it's this validation algorithm I was talking about previously. I'll try to go to this one here. So it's the first step is to divide the data into test data and then the inner segment, and then you subdivide it again into the validation and training segments. And then we're going to resample that inner thing to do first one PLS, rank the variables, remove the sort of crappy variables, do it again until we find sort of an optimal setting. And then we're going to resample all our data continuously. So we're actually going to remove the majority of features. So in the end, we're not facing 1,500 features, we're facing 45. And a lot of those are actually going to come from the same metabolite. Yes. So we reduce the 45 metabolites to only 45 features to only 10 metabolites. So in the end, we don't have to face a situation in which we do p-value adjustment because we only have 10.
And then those 10 you took in to look at the kinetics. Is that correct?
Yes.
Yeah.
That is correct.
Okay, very good. So I don't see anybody typing any questions. I'll just check again. No. I don't see. No. So the package for doing that, you said it's available in OR, is it? It's that MUVR package.
It's not available yet. We haven't published it yet. We're writing the manuscript now, actually. So we hope to release this into the public very, very soon. But there are, of course, other approaches to selecting variables. There's a bunch of packages in R. There's the Baruta package, the vSurf package. There's of course, you can do random forest modeling in itself to find, or you can do the VIP variable selection. But what we've seen so far from VIP variable selection, it's very, very dodgy. So I wouldn't recommend that to anyone, actually. However, the results from the Baruta and the vSurf package seem to be very, very reliable, actually.
Okay, very good. Okay, then if we don't, I don't see anything in chat, so I hope people see where we're doing it. I'll just type again to make sure people can see where I'm typing. Okay, if there's nothing else, then what we might move on. Not unless you have anything more to add, Carl.
No,
No.
I'm happy. Thank you very
Perfect.
much.
Well, thank you very much for your presentation and for your time. And I'll switch over to David now, who's joining us, as Carl said, from Canada. So David, yeah.
Am I online?
We can hear you. We can't see your screen yet.
Okay, we're just setting up here. Is that visible now?
Yes, we can see it. Yeah, perfect.
Okay, thank you very much, Lorraine, and also thank you, Carl, for a very nice introduction to time series. Good evening to everyone in Europe, and it is morning here. I'm going to talk about time series data using MetaboAnalyst. I assume some people are familiar with MetaboAnalyst, but others may not. So I'll have, I think, a brief introduction to MetaboAnalyst. And I think we'll see if we can move the... No, we can. Okay. I just have to... Okay. So MetaboAnalyst is a web server, and the URL is available at the top of that. And it's really designed to help with the processing of a wide variety of metabolomic data. You can work with NMR data, you can work with GCMS data, LCMS data, you can work with raw data that's been just simply spectral peaks or areas, or you can take lists of metabolites and their concentrations. Metabolites don't have to be identified, they can just be features. So as long as you can generate a table of data, you can do many of the things that are standardly done in metabolomics, from PCA to PLSDA to heat maps to pathway analysis and so on. I think we're just... so this is just a list of some of the tools that are available, and I've mentioned some of them already. And what I'll do, I guess, is sort of take a very simple approach, first of all, to time series analysis, and then I'll go into a more detailed discussion of especially ASCA, which Carl had already mentioned. So again, these are the different modules, and I've mentioned before you can take metabolite concentrations, peak lists, spectral bins, raw spectra. There's lots of tools available for checking integrity, to do some peak alignment, peak detection, data filtering, and of course with metabolomics data it's always important to do some transformation, scaling, and normalization. Once all that stuff, which is in red, has been completed, then you can do some of the statistical analyses and the power analyses and biomarker and pathway analysis. If you go to the website, you'll find the home page that looks like this. At the top, there's welcome, click here to start. It's almost invisible, and I've been complaining about it for years to get my programmers to make it a little more obvious. But there's constant updates, and the person who's been developing and maintaining this program is Jeff Shaw, and he's the one who also implemented the time series data analysis. If you click on the left side, you'll see a column, and there are things called overview, home, facts, tutorials, resources, update history. But then there's also something on data formats, which sort of describes how you have to assemble your data. And basically, it can take data that's in an Excel-like spreadsheet, and these are the types of data formats that it'll have. You can have compound concentration data, spin, bin data. Then there's time series peak intensity data, which you can have, and you can take a look at exactly how these things are structured, just to see how they're built or what sort of data formats you want. You can actually do time series data not using the time series one. You can actually just have compound data or peak data. So you're not necessarily bound to doing time series, at least that module, if you want to do time series data. So this is an example where you could just start off with statistical analysis, which is the basic module. These are ones when you click here to start, and you'll see statistical analysis, enrichment analysis, pathway analysis, and then there's time series analysis. But we're going to show you how to do time series or simple time series analysis just using simple statistics first, and then we'll dive into the more complicated one. So in this case, we're uploading some data, and it's asking for how it's formatted. In this case, we actually have concentration data, so we have tables of concentrations and metabolites. And this was, in this case, we've got samples in rows and concentrations in columns. The data set we have is, I'll call it a semi-synthetic data set. It's real data, but I've sort of recast it to make it more relevant to a time series. So real data with 9 dairy cattle that had a high cereal grain diet. And the rumen sampling wasn't exactly on different, well, yes and no, so that's why I'm calling it synthetic. But we have 4 sets of data that we'll call it day 0, which is before they start their diet, day 15, day 30, day 45. In this case, we're taking the rumen fluid from the cattle, and we used NMR spectroscopy to measure about 45, 50 metabolites. And the reason why this study was done is because it is known that high-grain diets, which are common in Canada for cattle, can be problematic, whereas grass diets generally are better for health of cows. So we've uploaded our data, and what it'll do in MetaboAnalyst is it'll immediately perform a data integrity check, and what you find is that there's 39 samples or sets uploaded, 47 compounds on average for each of the 9 cows that were measured over this time, and everything looks good, so we'll just press skip because the data was well done. And then what we're trying to do is we're both scaling and normalizing our data. This is just done to help perform better statistics, trying to make the compound concentrations a little more normal or Gaussian in a distribution. And you can play around with different ways of normalizing and scaling or transforming. And this combination of auto-scaling and using a pooled average generally created the best looking distribution, and this is just showing what it looks like. So here's the list of the 47 odd compounds before normalization, and you can see there's some compounds that are have very high concentrations and a lot of compounds with very low concentrations. And then using this approach for scaling and normalization, afterwards you can see that the distribution of concentrations looks a little more Gaussian or more normal. So this helps with the analysis. Now, because we're looking at 4 time points, we're going to do something which Carl had already mentioned, was just basically simple analysis of variance. And this is just to basically see, are there some temporal trends in what we're seeing? We could also do some simple pattern searching to see if there are, again, some temporal trends with certain groups of metabolites. Now, we could have done this time series analysis with just a single animal, and we might have measured the same animal 3 or 4 times as essentially technical replicates. But here what we've done is we've got 9 animals, we're measuring them once, and then we're measuring them over 4 different time periods on this one treatment effect. Now, as I say, this is simple time series analysis where we're not trying to deconvolve the effect of treatment versus treatment in time, and treatment interacting with time. We're just wanting to see, are there some trends over time? And this is technically time series analysis. So you can use ANOVA to do that. You can do pattern maps just to look at those trends, just to see if there's something interesting. So I've marked those different possibilities, and this is what you have when you start doing the data analysis. So as I say, we're looking at those time points, day 0, where before the grain diet is introduced, and then day 15 with the high grain, 30, 45. And then we're just trying to identify things that have changed, trying to say which metabolites are different between all groups or just between day 0 and everything else. And we can also do other kinds of ANOVA analysis. So when you clicked on ANOVA, and MetaboAnalyst is very much like a point-and-click system. we can see this compound, which is 3-phenylpropionic acid, and we can see at time point 0, 15, 30, and 45 days, you can see a trend in these box plots, which are in your upper corner, of things dropping over time. We can also see this plotted out where we're looking at the p-value in terms of significance, and it's now the log p. So it's 10 to the minus 7 or something like that in terms of significance, in terms of its difference between day 0 and the rest of the days, or between different pairs of combinations. So this is what ANOVA is very good at doing for looking at, rather than just cases and controls, it's looking at 3, 4, 5, 10 different groups, whether it's cross-sectional or temporal. You can, with MetaboAnalyst, browse. You can sort by P, log P. You can look for the Q value, and then you can click on different things. In this case, we're looking at uracil. We could have looked at LPS or endotoxin. We could have looked at 3-phenylpropionic acid or alanine. Those are all clickable, and you can see the trends over time. In this case, uracil is climbing over time, and that's a clear trend. And then you can see the standard deviations over these different cows. Now this is a temporal effect, and we're not distinguishing, the treatment plus time effects, and this is what ASCA is typically used for, but we're just simply saying, can we see temporal trends when we're looking at, hundreds of data points, which is often overwhelming. If we're looking at 2-time points, we can actually start using a tool called Pattern Hunter, where we can look for trends, not just simply are things climbing, but we can also look to see if something went up, stayed level, and then dropped, or started high and dropped and then went back up again over these 4 time points. So you could look for other models, whether if it's 10 time points, so you're getting a sinusoidal variation or a diurnal variation or something like that. So this is another tool that's available in MetaboAnalyst with Pattern Hunter. And you can see that on the left side where Pattern Hunter has been selected. And we're just looking, as I say, for linear, periodic, or other types of trends. In this case, all it's doing is trying to match the correlation coefficient with the particular trend. And here we have a linear trend that we're asking, where it's going at times points 1, 2, 3, 4, or 0, 15, 30, and 45. Does it go up? So in this case, endotoxin or lipopolysaccharide, glucose, and alanine go up. And the correlation coefficients matching this straight line, or a slope of 1, is around about 0.7, 0.75. On the other hand, if you recall, the 3-phenylpropionic acid, which is at the bottom in blue, has exactly the opposite trend. Instead of going up, it has a slope of -1. And it has a correlation or coefficient of minus about 0.8. So we could have looked for other patterns where we had, or encoded instead of 1, 2, 3, 4, it could have been 1, 4, 4, 1, which would have been high or low, high, high, low, or if it just went up after the first treatment, and so it went 1, 4, 4, 4, or however we wanted to describe it. And we will get these same correlation coefficients calculated to see if there's this linear trend. And as I say, you could do it for 10 time points, 20 time points, or just 3 or 4 time points. Another approach for just looking for temporal trends is just the heat map. This is a very visual thing that you can do with MetaboAnalyst. There's another tool we've developed called Heat Mapper at heatmapper.ca. Does the same thing. And you can look at individual metabolites over time, and you can just see very clearly what's happening. In this case, we just click on the heat map on the left side under the statistics package. We uploaded it. We've used a standard Euclidean distance measure. These are all the default values, and you just press submit. So this is a red-green heat map. Sorry if anyone's red-green colorblind, but what you're looking is at time, day 0, 15, 30, and 45 at the top. And I've just highlighted in yellow a set of compounds, butyrate, 3-phenylpropionic acid, acetate, isobutyrate, and so on, where in essence they start high, which is in red on day 0, and then they trend downward by day 45 or day 30 roughly to being green or lower values. Other clusters or groups which are different, but this is just a simple way of seeing temporal trends. Now it's not deconvolving treatment and time and treatment plus time or treatment times time. It's just simply a way of looking for temporal trends, and this is the basic or simplest form of time series analysis. And so we were just doing it using the statistical method. If you've been running this, it's been keeping track of all these things, and then you can ask for an analysis report, and at this stage it writes your paper for you and puts all the figures in. So that's a nice utility for a Tableau-Analyst. Now, we'll dive into the time series analysis next, and as I say, this is a little more complicated, and Carl's already introduced you to some elements of ASCA and the other tools. Like Carl, I find some of these things when you're looking at deconvolving time and treatment effects is a little obscure and probably too complex. And in this model, actually the time series model for us was originally done as a two-factor study, and then we said, well, we could just change this a little bit and we can turn it into time series because it's using a lot of the same sort of things. If you use the Tableau-Analyst time series module and not the statistics module, you have to make sure that there is a term time used in the group label. Versus the other one that I just introduced you to, which is the standard statistics, you don't have to use the word time. You could use anything you want. It's just simply looking for trends in the data. So just like what we did at the very beginning, you can upload your data. We've selected the time series module. We've chosen, in this case, a particular data set. It's an example set that you can upload. It's LCMS data. So the peak list or peak intensity table, and then we're looking at a Arabidopsis plant. We're looking at wound healing and we're comparing a wild type versus a mutant. And we analyzed over 4 time points, 4 days, starting day 1, day 2, day 3. And so this is what the data looks like. You can see WT for wild type, the different times, and then we're measuring the variables, which are basically these mass intensity values. When we upload, especially with mass spec data, we often do data filtering, and do usually apply an interquartile range filtering. It seems to help particularly well. If you've done this, as we did with this data, then we just press proceed, which you can't quite see on the screen, takes you over. This is just talking about why we use the data filtering specifically for mass data in Tableau-Analyst. And these are all just basically point and click, so I'm able to race through this a little more quickly. As with anything, we've uploaded the data, we still have to try and transform, normalize, and scale the data. So we're going to do a log transformation and a Pareto scaling on this data. That combination is seen to generate Gaussian distribution. You can see what it looked like before on the left side and after on the right side, and you can see that distribution looks fairly Gaussian. And then we have these paths that we can look at. We can do interactive PSA analysis, we can do two-way ANOVA, or we can do ASCA, or MEBA is another option. I'll just look at the two-way ANOVA quickly, and this is basically when you click on it, immediately it generates this interesting Venn diagram, and this is really how to show that it's assessing those components that Carl had talked about, where we're looking at temporal components, we're looking at, if you want, the treatment, or in this case a phenotype, and then the interactions between those. And so this is what these ANOVA deconvolution things do, is that they try and separate those components out. Whereas when I was showing you at the very beginning, we're not really worried about separating those individual components. We're mostly interested in just simply how has things changed with time. So this is diving a little further into the data and trying to control or identify some of these things that may be either just a consequence of time only or a consequence of the phenotype, slow healing, fast healing, or the interactions between those two. We can look at individual metabolites, or in this case these are features because we haven't identified them, and we can look between the mutant these 4 different time points, and the wild type at these 4 different time points. So the compounds don't have names like 3-phenylpropionic acid or glucose, they just have numbers in this case. But we can sort by phenotype, by time, or by interaction, the p-value, and we can scale and look for the highest and lowest values. In this case, we've looked for the one with the most extreme p-value under interaction, and we've selected this particular compound, and you can see where things are essentially going in opposite directions. At time 0, one is high in the mutant, and time 0, it's low in the wild type. So we could have clicked again on the time feature or on the phenotype, and again looked for the most significant in terms of p-values from this. So that's showing us some examples of interactions. The last part, which we'll talk about because we're running out of time, is just ASCA. And Carl's already mentioned this. And so this is, again, another method for looking at distinguishing or separating how time and treatment or phenotype and their interactions between those two can be deconvolved. It decomposes the variation into the data into individual variations induced by the experimental factors, time, treatment, phenotype, and those interactions. And this is, I say, where it gets complicated. And my own feeling is that sometimes it's overly complicated. And I think, as Carl pointed out, sometimes just trying to treat things a little more simply, you'll be able to find maybe not pulling out the specific kinetics, but you will see the trends that certainly you can talk about and interpret. So ASCA uses a PCA approach to do this, and if you clicked on the ASCA thing, with this data set, you'll get this thing first, which is a little hard to interpret, but if you just click on this tab called Major Patterns, you can see these trends. So between the mutant and the wild type, they are very different. And because they're only looking at 2 phenotypes, so fair enough. But then we've got 4 different time points, and we can also see a pattern in terms of the simple trends there. Fair enough. And arguably they're in opposite slopes. But then we can go a little further in looking at specific components. So it may not be an individual metabolite, it might be a combination of metabolites. This is where the PCA component 1, component 2, component 3, and so on. But you can see in terms of an component 1, we're looking over time at the bottom set, and we're looking at, I'm not sure if I can use the arrow here, so interaction between mutant and wild type. So in one case, this component, let's for sake of argument call it a metabolite only, starts high and is low in the wild type. That's dark blue one. Whoops. How do I go back? I'll do this. I'll be careful here. Just won't touch things, but you can see some trends. There's component 1 on the left, component 2 on the right. And they're very different between the mutant and the wild type. And you can see that explanation when you look at the time trend. So this particular, we'll pretend it's a metabolite. It's actually a component. The green one, which is the wild type, it starts off reasonably high. It starts falling, plummets at time point 2, and then rises back up again. In the mutant in red, exactly the opposite, the mirror opposite occurs. And so we can look at the time point 2 if we go up, which is the dark blue, and we can see, again, bottom left, the green and red are totally opposite, and that's mirrored in this interaction component 1 with the variation between the mutant and the wild type, where it's high on the left line with a slope of -1. The green line has a slope of 0, that's on the upper curve. And that's corresponding to time point 1. And so if we look down at the graph below, we see that there is no difference. That's the crossover point at 1, where we can see the green and red are overlapped almost in that lower graph. So the 2 trends are just plotted out in different ways. But this is just highlighting the different components, component 1, 2, 3, depending on how many PCA components are pulled out. But we could also pretend that these were metabolites. So this is pulling out or separating some of these things, as we talked about, which are time interactions, treatment interactions, phenotype interactions, which is what two-factor or multi-factor analysis does. It's also possible with time series. But it doesn't have to be this complicated, and so I really encourage people, if you're new to time series analysis, to think of the stuff that I showed at the very beginning of this presentation. And this just simply allows you to look for those temporal trends. It may not fully deconvolve treatment versus time effects or individual effects, but it certainly allows you to look at a whack of data that's sometimes confusing and to detect and identify statistically significant trends. So if you're wanting to learn more, there's two papers that have been written on this one. So one is on metabolomics 3, which has a very, very detailed set of protocols, and it's in current protocols in bioinformatics. And then the original paper describing these time series analysis and two-factor analyses was written in 2011. And you have, if you have really tough questions that I can't answer, which you may, Jeff Shaw is the guy to go to, and he's at McGill University, and he's always happy and willing to give very detailed answers by email or by phone. So thank you.
Thank you very much, David. I have one question, which is great. I think it's from Carl. David, what are your thoughts on using the ANOVA decomposition with supervised methods, such as PLS?
I think, I mean, in some respects, that's where the success that people have had with machine learning, so that PLS is, I call it almost a statistical version of some of the machine learning methods. So I think it's perfectly valid to do that ANOVA decomposition with a supervised method. Again, you need to have the appropriate controls, the holdouts and permutation analysis to make sure that you haven't overtrained your data set or on your training set.
Very good. Any other questions for David? Carl just replied that reflects his own thoughts, which is good. And so, David, you'd encourage people now. So people from FoodBall, because of the time series analysis we have, they will have complex data sets. So I guess your message is really to play maybe with the more simple approaches at first to get a feel for the changes in the data. Is that correct?
Yeah, exactly. Yeah, I mean, my view about statistics is it's the mathematics of intuition. And what we're trying to do is put things that, if we've looked at our data for a long time, to put our perceptions, our understanding of that data into a more formal, mathematically rigorous statement. And so, spend some time looking at heat maps, spend some time doing some very simple ANOVA analyses, which just, calculate averages and look at those different trends through box plots. And see if you're seeing something. If you don't see any trends, if nothing's changing over time, there's not a whole lot of time series analysis to do. And there's no point going on, to ASCA or advanced machine learning methods to see if there isn't any sort of variation over time. So yeah, I would encourage people just to use this simple statistical model, do simple ANOVA, do simple heat maps, do simple pattern searches, correlational analysis. This is classic time series analysis. And then if you want to deconvolve treatment versus time effects, then yeah, get into ASCA or some of the methods that Carl has talked about, which hopefully we publish soon. And then this will help tease out some of those more complex interactions, of which I think we're all interested in. But it's not worth going to great efforts when there really hasn't been any significant changes in time to what you're measuring.
Yeah, that makes sense. Yeah, and maybe I'll just ask one more question before we finish up. So in the ASCA on MetaboAnalyst, you can check, you were showing those PC components and how they separate, I think, on your last slide. So is it difficult then to actually go back to the actual metabolites that are making up those principal components? I guess it's as simple as in a normal PCA, is it?
Yeah, there's parts that I didn't have time to show, but you can look at some of these PC components. But I think, you know, this is a module that's not widely used, and I think if people are finding that they're not able to see certain components as they would wish, I would just encourage you to write an email to Jeff Shaw. He's been known to, you know, fix and implement things within, you know, 24 hours on special requests. And so if it doesn't give you that kind of information in terms of the metabolites, in terms of maybe a scores or plot, just email him and he can put that in.
Okay, so that's good to know, actually. So you can just email Jeff then. Yeah. I think Carl has just wrote another quick reply there. So he's just saying that individual variation may overshadow time effects in ASCA, or separation of individual variation can improve visualization and subsequent analysis. And I guess that's true. If you have small time effects, which in many instances we do in the context of nutrition.
Yeah.
Okay, just no other questions. I don't know, David, if you have any other comments, or you good?
Well, I just wanted to thank you for organizing this, and also for Carl to do such a nice lead into to this. I guess this is the very first webinar for FoodBall, and hopefully it won't be our last.
Yeah.
And again, I think we'll have other webinars on the subject.
Yeah, no, I think so. So I guess I would like to thank both David and Carl for taking the time to give make the presentations and give the presentations today. It's been very valuable. I think, as you said, it's our first shot at doing a webinar within FoodBall, and I think it has been successful. We have recorded it, and we will talk to Claudine and Jarly about making the recording available. And then, yes, we might look and see if this is a good mode of making presentations, and if we should think of other topics for going forward. But with that, then, I think I'll thank everybody and talk to you all soon.
Okay, thank you very much.
Okay, thanks, Millen. Bye.
Bye for now.
Thank you.